Hello, everybody. It's my pleasure to be here today. I will give a very general introduction. As Frédéric said, it's really the idea is to get all of you together with the next courses, with the next lectures that are going to be more specific. So it's just the idea to bring back from your memory some things about DNA, to introduce you to barcoding and DNA and meta-barcoding. So in the first part, I will really remember some basics about DNA, and probably for most of you it will be evident, but maybe for some of you it will be necessary. So that's why it's here. And the second part, I will focus on barcoding, DNA barcoding. And then in the third and last part, I will introduce you DNA meta-barcoding in order to introduce the next lectures that will be much more in deep in these topics. So all of you knows that DNA stands for desoxyribonucleic acid, and it's Watson and Crick in 1953 that has find the three-dimensional double helix structure of this molecule. But what interests us here is that DNA is present in all living organisms. All living organisms store their genetic information in DNA, and that's the main point for what is going to follow. How do they store this genetic code? It's through an assembly of 4 nucleic bases. You have A, all of you maybe knows A, T, G, C. So A is for adenine, T is for thymine, G for guanine, and C for cytosine. And these bases, nucleic bases, goes by pair. And you have A that goes with T, and you can see on the right, on the top, you have A and T, which are linked with 2 hydrogen bonds. And so they are linked together, and G and C are linked with 3 hydrogen bonds. And that has some importance, because that means that the link is stronger between G and C than between A and T. And Clarisse may go further on this topic. Here it's another representation. You can see that, for example, on the top left, you have C, the C base, which is linked to, in gray, sugar, and also to a phosphate group in the red circle, yellow red circle. And this altogether, base plus sugar plus phosphate group, is called the nucleotide, and nucleotides goes by 2 and assemble in this double helix. And the strength of this double helix, how is this DNA organized? It's organized with coding regions, which are called exons, and with non-coding regions, which are called introns. And as you can see here, coding regions, well, maybe it's not so easy to see in this figure, but coding regions are much smaller than the non-coding regions, which are the vast majority of DNA. So how does it go to have the transcription between the DNA to the protein? So from the DNA, the double helix in the nucleus, you will have this in RNA in a single strand RNA, and that will be submitted to splicing in order to keep only out of the nucleus in order to be translated into protein. So we have DNA in all living organisms, but some things also is that all living organisms share similar genes that are inherited from a common ancestor. And you maybe know this draft from Darwin in 1837, who has this first intuition that there was a link between of a common ancestor with all living organisms. And with genetics, it has been possible to ascertain that. And here you can see now that we better know how is organized this tree of life with 3 main domains: archaea, bacteria, and eukaryota. And so, yes, in fact, similar genes are inherited from a common ancestor. We all have similar genes, all organisms, but all organisms have a different genetic code. And this is important because that can be used as an identification tool. That's why DNA can be used as an identification tool. So that leads me to the next part about DNA and barcoding. So as I said, DNA is can be used as an identification tool because it's in all living organisms, in all their cells, and even in all their cell remains. Here, for example, for us, for human, you can see that we have DNA in any part of our body. And you probably know that it has been. Okay, so the second part is on DNA and barcoding. DNA, in fact, can be used as as an identification tool because it's in all living organisms and in all their cells and remains. As we have DNA barely, we leave DNA everywhere, so that's why it's used in criminology, and because when DNA is found on a crime scene, it can be compared to the DNA of different suspects, and then by comparison, you can find if the criminal is among your suspects. And for example, here I don't think you suspect 2 has a similar DNA as the one that was found on the crime scene. Okay, so the idea of using DNA as an identification tool drives Paul Hebert to the idea of DNA barcoding. That's the idea that each species have a specific DNA. Part of this DNA may be used as a barcode to. So that comes in the publication from Paul Hebert in 2003, who make the proof of concept of using DNA barcodes as a biological identification tool. Is it okay now? Yes? Okay. So his idea was to use a mitochondrial gene, cytochrome C oxidase subunit 1, with a barcode region, not so short, in fact, of 658 base pair region of this gene that could serve as a core of a global bioidentification system for animals. So that was his hypothesis. And he wanted that for identification, but also because he was observing that there was more and more lack of taxonomical experts, and that if we want to keep this expertise, that could be a tool for sharing taxonomic expertise. And he also promoted DNA barcoding as a standardized method that could use the information that are in all species at all their life stages, including early life stages like eggs, for example, and in all their tissues. So how does it go? First, you take a specimen. So if you want to take a tiger, try to take a sample from that specimen and extract the DNA from that sample. Then you amplify the selected barcode with PCR. And now I think everybody after COVID knows what is PCR. So polymerase chain reaction, which allows to amplify a selected part of DNA. And then you sequence these small amplified barcodes in order to have to read the DNA sequence. That's why we call that reads. And you compare this read to a reference database where you have the link between a DNA sequence and taxonomy identification. And then you can obtain an identification from that. So he proposed the proof of concept on insects. He was a guy working on butterflies. He is a guy working on butterflies. And he make a reference database with 100 families from 8 orders and with 1 specimen per family. So each specimen, so 1 specimen per family, was identified taxonomically and also with morphotaxonomy, and he sequenced the COX-1 barcode. And so he could make this reference between DNA and taxonomy. And he looked at the taxonomic classification. The morphological based one was consistent with the DNA based classification. Then to test that, he took in 50 specimens that he didn't know, that was not identified previously with morphotaxonomy, and he sequenced the COX-1 barcode, and he was able to correctly assign all specimens at the order level. So that was his first proof of concept. Then he went on a more challenging test on Lepidoptera, because as I said, he was a butterfly guy, and because it's a challenging group, because there's little polymorphism in the COX-1 barcode. What does it mean? It means that in this barcode, there's not so much differences from one species to the other. That means that it may be a bit difficult to disentangle between species. So he had a reference database. He made a reference database between 200 species, which have a correspondence between DNA and taxonomic identification. And then he made a test on 150 specimens that were not identified previously. And he was able to, in fact, correctly assign all these specimens at species level by comparing the DNA sequences he obtained to the reference barcodes in the reference database. Here you have a small subset of this these specimens. You have the 18 species from RTCD. In the graph, the cross stands for the specimens that were to identified, and the circle stands for the references that were already in the database. And when you see a cross and a circle that are together, that means that the test, the cross, was correctly identified to this species. So, for example, on the bottom, you have this. I cannot see the name very well. This Cycnia oregonensis, where you have this cross and this circle that are very close. So it was correctly identified. But as you may see also on the upper left, sorry, there is 2 Hippophrepia species that are very close to each other. You can see that the 2 circles are very close to each other. And but still the crosses could be assigned correctly at the top species, the Hippophrepia fucosa. And but from that, you can see that probably the identification was not so easy for this species. It was more obvious for some others. So just to make you enter in more detail in what it means, and Clarisse will give you more detail on that. So after this proof of concept, it was really this idea to have a standardized taxonomic approach based on DNA with a small fragment, easy to amplify, that was used as a barcode. But this is what is important for that, is that there is a barcoding gap. I will define that now. Here you have 2 species of diatoms. Each of it has its own barcode, its own sequence, DNA sequence for the barcode. And if you sequence different species, different individuals from the same species, for example, species 1, you will not obtain exactly the same sequence. You will obtain slightly different sequences with some genetic distances, which are figured here by the red arrows. And if you sequence different specimens from species 2, you will also have some differences between the individuals. But what is important is that the distances between the different individuals from each species is always higher when it's interspecific distances than when that's the yellow arrows, than when it's intraspecific distances, genetic distances. And you can see that also on this graph. On the x-axis, you have the genetic distances. So on the right, you have the distances, what is figured by the yellow arrows. So you have the interspecific distances, which are higher than in the red, the intraspecific distances, which are the distances between individuals from the same species. And the gap between these two kinds of distances is important to ensure that you have a good identification tool. But this is not always the case. And sometimes you have this overlap between genetic intraspecific distances and interspecific distances. That means that in some cases, you may have intraspecific distances that are higher than interspecific distances. So it makes the job a bit more complicated sometimes. So I will give you an example to see that more in deep. So for example, here you have an example on spiders by Kandek and Kantner in 2015. They sequenced barcodes, COX-1 barcode, for 1,203 individuals that were from 162 species that were from 3 families. So Araneae, Tetragnathidae, and Lycosidae. And they could see that there was a good efficiency of the DNA barcode for the identification of spider species inside each of these 3 families. So for example, in this graph, you can see on the y-axis, you have the genetic distances. And the first box plot is for Araneae intraspecific distances, while the second one is for Araneae interspecific distances. And then you have the same for Lycosidae, and then the third set of bar plots is for Tetragnathidae. So you can see that there is a difference, indeed, between the mean intraspecific distances and the mean interspecific distances for each of these families. But as I will show, it's not exactly the same in all the families. You can foresee that from the bar plots, but this, you will see it more in detail. So if you look at the, for example, in the bottom left, you have the Tetragnathidae family, and you can see in blue the intraspecific distances between individuals from the same species, and in red, the interspecific distances between different Araneae species. And you can see that there is a clear gap. Well, okay. But if you look at the Lycosidae at the top right, then you can see there is also a clear gap, but you can see that the distances on the x-axis are very different. For example, the mean interspecific distance is here about 6, while for Tetragnathidae it was something like 17. So you see, it's not organized in the same way in these different families. And if you look at the Araneae, then it's a bit blurry, and you cannot make, there is not this nice form of the two distributions. And if you take all them together, that's on the right and the bottom, you can see that, well, there is a strange organization. And you can see that the size of the barcoding gap, in fact, depends on the taxonomic group. So this is an explanation, but still, you could have good efficiency, but you have to take that into account. And also, one of the dream of Paul Hebert that it was that this DNA barcoding identification tool could be used with a single barcode. You could identify all living organisms on Earth. That would be a universal barcode, but in fact, it has been soon obvious that it was not possible. Of course, COX-1 works well for most metazoans, but for fish, for example, there is another barcode that is more efficient. For fungi, there is another one. For plants, for diatoms. I will not go in detail here because Clarisse will go in more detail in a few minutes, and also Frédéric will go in very much detail for phytoplankton and for diatoms. So this is not the point here, but still I want to show you a study that we did, well, maybe, yes, more than 10 years ago, testing different barcodes that were available for diatoms. In a diatom cell, you have the nucleus, where you could find the nuclear gene 18S, which is coding for the ribosome RNA 18S. You have mitochondria, where you can have the COX-1 mitochondrial gene, and you also have, you can find the RBCL, which is a chloroplastic gene, coding for the large subunit of the Rubisco, which is an enzyme used in the photosynthesis. Well, we wanted to compare all these potential barcodes and see if they can help us to have a better knowledge of some species, in fact. And we did an experiment on one species, Gondula, is probably online today. So hello, Lenaïque. And she isolated during her PhD 25 strains of Gomphonemma parvulum from tropical streams, but also strains from temperate streams, and also different Gomphonemma species streams that were isolated also. And we compared the sequences for the 3 different barcodes that I showed you just previously to the taxonomy identification based on morphotaxonomy. And she did a really in-depth morphotaxonomy analysis here. So what we could see is that the 18S, the nuclear gene, was really great, allowing to clearly identify the Gomphonemma parvulum inside the Gomphonemma genera. But the other 2 others, COX-1 and RBCL, were able to define really clear place inside the Gomphonemma parvulum. So it shows us that it was a complex species with cryptic diversity, and that was interesting, and that helps go in deep in the taxonomy. I will not go here in deep. So from what you've seen up to now, you could see that DNA barcoding, it really has the crossroad of different disciplines. Of course, you have the wet lab part with the molecular biology. You have the taxonomic part, because you need to have strong reference databases where you need to have clear taxonomic identification. And you have the informatics, bioinformatics part, which is also very important to make sense from the sequences. And this collaboration between taxonomists and molecular biologists is very important for something that is a key point of this DNA barcoding methodology, which are the reference libraries. because end users will use these libraries to identify sequences that they may get. And if they are not strong, accurate, then I would say DNA barcoding is useless. So you need really a strong collaboration for taxonomists, between taxonomists and molecular biologists to really ascertain the taxonomic identification of barcodes. And for example, here you have the library, which is called BOLD, developed in Canada by Paul Hebert. Here, for example, for how does it work? For one butterfly specimen, you have the specimen, you keep it in the collection. You have all the metadata that goes with the specimen. That means all everything about where you collected the sample, who collected it, who identified it, and so on. You also have photos of the specimen details when you need some details to really identify it precisely. And you also take a tissue sample from the specimen in order to do the DNA part. So to obtain the DNA barcode with DNA extraction, PCR amplification, sequencing, so that you can also store the DNA barcode in this library. And then you can do that for many species. And then you can build it. It's built as a reference library where you can really make queries where you can enter sequence, and when you will get a taxonomy from that entrance, from that query. But you probably, those of you who are knowing more microalgae, understand that it's not very easy to take a leg from or a tissue sample from a microalgae. So for microalgae, building reference databases is quite different, because from one, you cannot, in fact, most of the time you cannot from one cell have both taxonomic identification and obtain the DNA barcode. So you have to go through this strain isolation, strain culture, and then from the culture you will be able to take photos to identify the specimen, and you will be able to extract DNA from part of it. Then in your reference library you may also store the culture, frozen culture, living culture when you can, and also the DNA. So it's a much more complicated job to do that. But Frédéric will explain you that there is also some other way to improve reference libraries that will bypass this isolation and culture step in order to strengthen the libraries. So DNA barcoding, which uses? You know them probably. With a barcode, you can identify endangered species. Probably for this kind of animal, you don't need really a barcode, but well, that's the idea. You can also identify invasive alien species, identify sensitive species. What I mean by sensitive is that species that could be of commercial use or of biomedical use or of interest because they are pathogenic, and so on. And in the environment, you may also be interested by key species, indicator species. So all these can be identified through DNA barcoding for different purposes. That could be quarantine inspection, fraud control, but also environmental assessment, which is more the topic today, and biodiversity conservation. Something that is very interesting also for barcoding is that you can use it to identify unidentifiable organisms. That means sometimes you have incomplete specimens that are degraded, or you may have only a part of a tissue, or just a fragment, or it may be incorporated in food or whatever. Or you may have only young stages that are larvae or eggs, for example. And then you can using DNA barcoding, obtain an identification from that. That's very interesting. And also another big interest is that it may give you, as I shown for this Confonema parvulum, for example, complex, it could help you to better describe diversity, access to undescribed diversity, access to cryptic diversity. Okay, so this drives me to the third part about DNA and metabarcoding. Yes, you have some questions. Is there some online questions? Okay. Yes, it is indeed. I'm sorry, maybe all of you are sleeping. I'm afraid. I hope not. Okay. So maybe figure your question for the end of this talk and we can discuss that in deep. So from this first approach of Paul Hebert in 2003 about DNA barcoding that has been then much developed, the idea of this standardized DNA tool for identification with a short barcode that is easy to amplify, short, that is taxa specific, and so really something that can be standardized. The scientists went to a more global tool of identification at community level, which has been named by Taberlet and collaborators in 2012, metabarcoding, DNA metabarcoding. So this is an extension of the concept to the study of natural samples, where you will have a mix of different species. You will have community samples, and you will need to identify all the organisms of the community, within a community. And for that, to use new technologies of sequencing, high throughput sequencing, what we called also next generation sequencing, in order to have a good representation of a community sample. Here, for example, so barcoding at the top, you have one culture, you want to identify it, and you have one barcode that is related to it. But in an environmental sample, you may have a mix of different diatom species, for example, here. And each of these species have its own DNA barcode. And the idea is to identify all the species inside this sample. The benefit here is really to reduce analysis time, to reduce cost, because it's a high throughput method, and also reduce cost, but that may mean also keep same costs, but you analyze more samples. And also it's a way to facilitate intercalibration, because here we will have a tool that will be based on DNA sequences, while on morphotaxonomy, you often have, it's often depending on the expert that is analyzing the sample, and on his expertise, on his references, the books he used to work with. And sometimes there may be some differences between the results between two different experts. Here you have something that is straightforward, and that could be intercalibrate from one experiment to the other. So the idea on the top, you have barcode, so you extract DNA from one organism and you try to identify it. And at the bottom, you extract DNA from a mix of different organisms, and you try to have a clear identification of each of them. So it goes a bit like the workflow I showed you for DNA barcoding. That means that you have these different steps. You have the environmental sampling. Then you, from this sample, you will extract DNA. You will amplify the group you are interested in with a specific barcode that is specific of this group of interest. Then you will go through ATS, that stands for high throughput sequencing, to obtain the reads of the different organisms that are in this environmental sample. And through bioinformatic processing, you will try to identify the species that are composing this sample. So in fact, it's three main steps. You have a first step, which is really field sampling. You go in the field, and which is not very different, which is quite the same sometimes, or not so different from what you used to do with morphotaxonomy identification. Then you have this wet lab part where you have to have a molecular biology facility. That includes the DNA extraction, DNA amplification, and sequencing. And then you have the bioinformatics part, where you make sense from the sequences you obtained to try to make a floristic or phonistic inventory. What's interesting also with DNA metabarcoding, that has not only you will have one sample with it, which is a community, so you have different individuals, different organisms in it, but also in a single sequencing experiment, you may multiplex, you may add different samples. You will give label to all of these all the different samples, and you can process them all together in a sequencing experiment. So that makes really a high throughput method where you will have both. You may have both high sequencing depth per sample, so a lot of information per sample, but also you can have that for numerous samples that are analyzed together. For example, currently we analyze 380 samples together in a single sequencing experiment. So what's the material to do this metabarcoding, DNA metabarcoding? So you have biological DNA, which is in the organisms, in their tissues, in their gametes, in their cells. But you also have DNA that is particular DNA, which is in the organelles, which is just protein complex. And this can be inside a cell, or this can be outside a cell. And this can be cellular DNA or extracellular DNA. So indeed, the organelles, the protein complex, this particular DNA may be also extracellular DNA. And for example, in aquatic ecosystems, you may find them in the water. You may find them adsorbed or on different components or dissolved in the water. So that's the basic material that you will use to do DNA metabarcoding. So that brings me to the definition of environmental DNA, because there is a bit of discussion between experts, because what we call environmental DNA in the broad sense is all DNA in environmental samples, in all environmental samples. That means that includes all these definitions. But you will sometimes meet people that have a much smaller definition, much precise and strict definition of eDNA, which is only extracellular DNA in environmental samples. That means only free DNA that you may find, for example, in water, or free DNA that you may find in soil. And to my sense, to our sense, well, it's not so easy to disentangle between both, because if you want to, for example, to take samples from phytoplankton, you will have both free DNA that is in the water, but you will have cells, which is intracellular DNA. If you take biofilms, you will have diatoms in these biofilms, but you will have also some free DNA in the biofilm matrix. And so, well, just to know that some people are very strict and don't want you to speak about environmental DNA when it's cellular DNA, but just be aware of these two definitions. So now if we go to the uses of DNA barcoding, meta-barcoding, so you can take samples from many different environments. Of course, here we are more here to speak about aquatic ecosystems, but you can also take samples from honey, for example. You can take samples from soils. You can take samples from feces. You can take here in aquatic environments from it. You can take samples from biofilms and probably many others that I have not presented here. And what do you do with that? After amplification and sequencing, you obtain a taxonomic list, and you can assess plant diversity, for example, with soil samples, where you will have the roots of the plants that are living in this environment, and you can assess plant diversity, for example, from that. You can also assess pollution responses. For example, if you sample, would say, for example, phytoplankton in different aquatic ecosystems that are exposed to different pressures, for example, more or less pesticides, more or less herbicides, and you may assess the pollution response of the algae community to these pesticides. You can use that to make diet analysis, to analyze the trophic relationships between organisms. You can use it in ancient ecosystem. For example, in the aquatic ecosystem, you can take sediment cores where you could really assess the organisms that were in the different layers of the sediment core. If you want any details on that, you can ask Benjamin, who it was his PhD topic. And you can also try to see if there is some invasive species that are inside your community, or you can be interested in using the taxonomic list to to calculate biomonitoring indices. So just to focus on aquatic ecosystems, you can have many uses. Here you have, for example, this picture show you that in the water you can find DNA from different fish species, and that you may find the composition of the fish community in one environment using water and free DNA in water. In that case, it's really generally eDNA as a switch sense. But with DNA approaches, you can really do many things in aquatic environments. That goes from species detection, of course, again, invasive alien species, endangered species, but also parasites, pathogenic species. You can use the tool for biodiversity survey of different groups, mollusks, macroinvertebrates, frogs, whatever you want. And you can also use for the tool for to calculate the values of some biotic indices using biological quality elements that are used for the biomonitoring of aquatic environments. And that will be more the topic today and tomorrow. And so to focus on that topic, that's the usual workflow that goes from the field sampling to the sample preparation. So of course, it's different if you have macroinvertebrates or if you have algae. You will prepare your samples differently. Then you will have your expertise, your references, your books to identify and to quantify. So for microalgae under the microscope or under the binocular for macroinvertebrates, to quantify the list. So you will have a list of with a semi-quantitative list of species that will allows you to calculate an index value, and that will give you, according to the value, an ecological status to the ecosystem you have sampled. But with DNA, it's about the same, but the steps are a bit different. So you will, your sample preparation is a bit different because you will extract DNA, you will amplify the barcode you are interested in, and you will sequence it. And then you will have the data analysis step, which can refer to reference databases, but that can be also a bit different. And Frédéric will show you a different way of analyzing this data, which relies more or less on references in order to calculate index values. So, for example, here for the items, which is among the biological quality elements that are required for the water framework directives, WFD in Europe. You can sample from the biofilms, you will extract your DNA, you will amplify the barcodes that you are interested in, and you sequence this mix of different amplicons, we will call amplicons, that is the barcodes that are amplified. And you will analyze the reads you obtained by comparing it to a taxonomic, to a reference library, where you will associate your barcodes to species. And you will have a semi-quantitative list of taxa. So you see a species with an associated percentage of abundance, associated abundance in the sample. And this will allows you to calculate an index value. So Frédéric will go in detail on the way to assess the quantification, because this is also a bit special, because it's much different from what we do with microscopy. So you will obtain taxonomic inventories with relative abundances, and from that you can derive quality indices. Just to show you very briefly, this is not the topic here, but that there is also other DNA approaches other than metabarcoding that you can do from a DNA sample, a mix of different DNA that you have extracted. You can process metabarcoding, of course, but you can also have probes hybridization or shotgun sequencing. And each of these approaches have its advantages and drawbacks. And for example, for metabarcoding, I think it's very interesting because the data treatment, as you will see here and later maybe in another training, is quite easy to do. It's, as I said, it's also cost efficient because it's a high throughput method. But there is this PCR step which brings some biases, and Clarisse will probably go in some detail on that. And also it's dependent on the primers and the barcodes you are going to use. And because with different primers or different barcodes, you may have some different results, more or less different results. The other methods have also some interest or some disadvantages. For example, these two methods are not using PCR, and so there is no distortion of the DNA signal, and so that's an advantage, for example. So metabarcoding is one of the many tools that you may use with genetics. You also have metagenomics, metatranscriptomics, metaproteomics, metabolomics. And each of these tools has specific application. And here we will focus on metabarcoding, and which is more dedicated to analyze species and genetic diversity. while some other applications are more dedicated to understand better ecological functions. Okay, so with that, I think I'm done. I thank you for your attention, and I'm ready for questions. And I don't know. Thank you. Is there any question? Yes.
Oh, Alexandra Markovic, ICTM. Thank you for the great lecture. I think I have actually too many questions in my head, so so I won't ask too much at the point. I wanted to ask, as the organizer, will the participants get the presentations? I guess we will send them the presentations or not.
I think we will send them the recording.
Yes, the recording
Yeah.
with the presentation. Okay, so the participants will get the recording of the lecture with the presentations for you to know. And I wanted to ask, everything is quite new for me, but this high through output sequencing, this is actually, how to say, if you are barcoding, then you use PCR, and if you are doing meta barcoding, then this is like an analog method, or right, to amplify the sample, or?
Yes, yes. In fact, you will amplify it the same, but it's just your first sample that is different. When you do barcoding, you will try to identify one species, so you will have one species sample. Sometimes you may have a more complex sample, but in which you want to find one species because I don't know, food, and you don't know if they use this type of fish, for example, in food, and you want to identify it, then it's barcoding. But and when you go to meta barcoding, you have an environmental sample. You want to really assess the community composition of your sample.
The purpose is to amplify the sample, right?
Yes, yes. In both methodology, you extract the DNA and you amplify a barcode, and you identify this barcode. In one case, you have a single barcode, that is barcoding. You will get a single barcode, and you will compare them to a reference library and say, okay, this barcode is really this individual, this species. And in the other case, you will have a mix of different species, and you will say, okay, in this mix, I have this percentage of this species, this percentage of this one, and there is some I cannot identify, and so on.
Okay, thank you. Thank you so much.
I have some questions from Zoom participants. What is the difference between DNA metabarcoding and eDNA metabarcoding?
Well, it's again, it's the difference is the initial sample. In fact, if you take well, as I say, it depends on the definition of eDNA you use. But if you use eDNA in a strict sense, that means that okay, if you use eDNA in the strict sense, that means that you have a mix of DNA in a sample, and you want to assess from that sample, that mix of DNA, the relative percentage of all the species that are composing this sample, and this is DNA metabarcoding, and it's not so different. Would you add something, Frederick?
Yeah. In fact, I really enjoyed your picture where you have this gradient of different kind of DNA, from cellular DNA or fragment of DNA or DNA in organelles, and then you have extracellular DNA. Yes, maybe we can come back to this picture. Yes, this one. Yeah, I think it's really interesting. Just maybe to answer to the person on Zoom. In fact, when you say what is the difference between DNA metabarcoding and environmental DNA metabarcoding, we could add what is the difference also between cellular DNA or particulate metabarcoding. Sorry, what is the difference between cellular DNA metabarcoding, particular DNA metabarcoding? So, and we also talk about massic DNA. I don't know if it's purely French definition. Massal DNA. It's an additional definition about DNA, which is really in the body of organism. Yeah, but yes, in fact, there are many definitions like that. I think it's good to have this nice picture from Tristan Lefébure, a good colleague from us in Lyon, and this gradient of different kind of DNA, really from the body of the organism until the DNA, which is really free floating in the water.
Also, muscle DNA is used for macroinvertebrates. Maybe some of you knows about DNA for metabarcoding for macroinvertebrates, and usually you have a mix of the organisms. It's really the body, as Frederick said, the body cells that are used and mixed and extracted to obtain the basic sample.
Thank you. Thank you, Agnes, for a very nice presentation lecture. I would like to ask you about the quantification of metabarcoding samples. You know how to do it, classic taxonomy and classic analysis in labs, how we have a community.
Yes, I can say briefly, but I think Frederick is really going to go in deep in that in the next step, because in fact, it will be a bit different according to the organisms you are going to analyze. Well, the main feature maybe is that the bigger the organism, the more DNA you have. So that may so if you have one big organism and one small, you will have a DNA signal that is proportional to more or less to the biomass, let's say. So you will not. So it's not a direct quantification of how many individuals you have, but it's more linked to biomass. But Frederick will show you a way that we proposed, for example, for diatoms, to go back with this biomass to individuals, and really to have a good evaluation of the richness. And yes, but that is something that is not easy, of course, especially when you go to, for example, fish, where you have free DNA in the water, which the presence of this DNA will really depend on the phenology of the species. So when it's a reproduction time, for example, they will release a lot of DNA in the ecosystem, while at some time of the year they will not release very little DNA, only very little DNA. So well, there is some way to do, but here we will focus on the way to do it for diatoms, and we will speak about it also for phytoplankton, although it's not so easy, in more details tomorrow, this afternoon or tomorrow, I can't remember.
I'm not sure if the question is for you, Agnes, or maybe Benjamin could help us, because it's related with sediment sequences. I would like to ask from how old sediments we can perform the metabarcoding technique. For example, is it working for around 1 million years old sediments?
I don't know, but I don't think so. Benjamin, you have some ideas?
Just a second.
I know that 100, 200 years, yes, it's fine.
In fact, I work on time period from 200 years ago, and but I think that you can use metabarcoding for time period of 1 million, but I never work on this time period. But in fact, there is a taphonomic process which degrade DNA. So more you have a long time period, more DNA has degraded.
Frédéric, you want to say?
Yes, and it really depends on the matrix where you are. Okay, Clarisse agrees. If you are in sediments which are with no oxygen, so the DNA will be more constant. And if you are, for instance, in the biofilm, for sure, it's always with lots of oxygen, and there is a oxidization, which is very quick. And there are a lot of bacteria which are ready to eat all these DNAs. And so here, using DNA for a long time period is not possible. So yes, it really depends on the matrix where you are.
And it also depends on the organisms, because some are not protecting, the DNA is not protected and will be very quickly degraded, while others you will find it. So you want to add something, Benjamin?
Just for example, if you work on resting eggs of a cladoceran, for example, the DNA is protected by a capsule, and so you can use this matrix, but in free DNA, it's more degraded by environmental factors.
Okay.